In today's episode of the Human Equation podcast, I'm joined by Professor Marla Feller, a distinguished professor of biology of biological science at the University of California, Berkeley, and a member of the Helen Wills Neuroscience Institute. Professor Feller studies how the brain begins organizing itself long before we are born and aware of the world. Her work focuses on the retina, where she investigates how early spontaneous activity patterns, known as retinal waves, shape neural circuits that later support vision and perception. Rather than studying vision as something that simply turns on at birth, her research reveals how early neural activity prepares the brain in advance, essentially giving the visual system a practice run before real sensory experience begins. She's a fellow of the American Association for Advancements of Science and a member of both the American Academy of Arts and Sciences and the National Academy of Science. We are honored to welcome Professor Feller. Thank you so much for agreeing to come on this podcast with me. I'm super excited to speak to you for many different reasons, but the most just your mind and your work and the way that you've done research in the past. So thank you so much for coming on.
Thank you for the invitation.
Yeah. So I wanted to start off the conversation with certain questions, but then I was having a conversation yesterday with another scientist, and so I wanted to get your opinion on what you thought about that question that we posed or to each other. Do you think that AI will eventually replace scientists, right? Where we'll have AI agents that are scientists. What are your thoughts on that? Like, do you think that, we are in jeopardy of becoming extinct?
Yeah, no, we're all worried about being in jeopardy of becoming extinct for multiple reasons. But I am not as worried about AI. I mean, I think what AI can do is certainly help us in science because it's much better at curating the large amounts of data that's out there, making sure we're aware of all the studies that have been done in the areas that we're interested, that we're doing research in, so we're not as likely to duplicate studies. There's been a lot of really wonderful advances in kind of curated large language models that have really helped in clarifying your logic that you're using, and things like that. But it cannot, I am one who believes there will not be this kind of generalized AI. I don't think that they're going to be able to think creatively. They may come up with some really interesting hypotheses, and I think based on its reading of the literature, which will be more thorough than a human can do. Yeah. So I think it's going to be wonderful for science. So that's my hopeful take on it. I am not a hopeful person, just to be clear. I'm not a glass half full person. But for this, I'm feeling more positive.
That's a very orthogonal response that I wouldn't get from the scientists that I speak to. A lot of the response is a little bit more adversarial. But I'm hopeful. You've made me more hopeful, right? Because I think, indeed, right, like being able to clarify or go through literature that you can't as a human being by virtue of your scarcity of time and being able to connect dots in ways that you can't because you don't have all infinite perspectives as, let's say, the agent does. And so hopefully, there could be some integration. I know in the tech space, which I'm sort of in as well, I found that a lot of startups don't really serve academics because of their unwillingness to pay for things and the products, right, that are often, right, developed. And partly the way that scientific funding and pay is, people normally are not really driven by capital, but more of other things, right? And so there's a lot of different nuances there. With that said, what are your general thoughts on the state of academic funding right now? And sort of the current administration gutting, funding that the lack of grants, on top of the other things that we as academics and scientists face with publication and having to pay and things like that. What is your general thought on that?
I mean, this to me is the extinction risk. So there's so many ways that this administration has been dangerous to science. So there is the limitation on funding, right? So it's a very stressful time because no one really knows what the rules are right now, to compete for the funding that's there, because the rules keep changing.
Right.
So that part is, definitely we can't do it without funding, or we can't plan if we don't know what the funding is going to be, right? We are very adaptable, but we just kind of need to know the rules and the landscape so we can plan accordingly. I think, and then two other areas where I think it's been devastating is this breaking up of international collaborations, making it very difficult for international scientists to work with, to come to the U.S., to work with us, and for us to have free flow of ideas.
Yeah.
That international scientists are really hesitant. They can't come to conferences. They don't want to come to conferences. They feel very unwelcome, by the administration. They're scared, and so that has been, so that's a big hit at the intellectual level, because science is really a very international enterprise.
Right.
And then I'll say the third, I can talk about all of these, but the third one is the politicization of science.
Right.
And that is the part where I feel like my head is going to explode, and I think that's where, they all are upsetting, but that's to see which facts we're going to pay attention to and which ones we're not going to pay attention to is just so orthogonal to the way scientists think about everything. You want more data, you want more information, you want to make decisions from an informed place. And in my mind, I go back to this, something that Trump had said in his first administration is, if we stop testing for COVID, then nobody has COVID, right? If we stop getting data for climate change, then we have no problem, right? If we stop, and that, if we ignore studies on how vaccines save lives and enhance studies that or promote studies that the scientific community have rejected and saying they're the gold medal standard. It's just like, why would you do that? Like, I can't. So that part has been really hard to take, and that has real consequences for human health.
Right.
So crazy.
So with that said, do you think that we need to start thinking about new funding mechanisms? Because, for me, I, run a startup, but I'm also a scientist. And so the convergence of the two feels very much like a possibility for me, right? But of course, right, like not most scientists don't run a startup, right, while they're doing science. But we have so much to offer as far as the knowledge and the innovation that a lot of scientific research curates. And so do you think that there could be a new funding structure that emerge? Because I also see this as not just as a concern, but as a moment of opportunity. What do you think?
Yeah, and I think in my world, in the world of neuroscience, there are definitely some privately funded neuroscience. There's the Howard Hughes Medical Institute. There's the Simons Foundation, which funds a lot of different areas of science, but they do research in autism. They fund research in autism. Sergey Brin helped start this organization called ASAP to study Parkinson's. And I think those are all great. Even though they are very well funded, they pale in comparison to sort of what the NIH funds and NSF. So there's that. It's and I don't know who decides what we're going to work on, right? So if a very wealthy person, I guess Sergey Brin is worried about Parkinson's. I think his parents have it. He might have a genetic disposition maybe to get it. So he's very interested in it. I don't know if any of that's true. I'm assuming that that's why he's interested in it. And that's great. But and that's great for that field. But is that how we're going to decide kind of what science is going to be done, or do we want it to be a little bit more bottom up, which is the way that it's been done traditionally? And it could be, I'm doing all of my funding comes from the National Institutes of Health. I am well aware that it's publicly funded, and so the public has to decide, if they want work to be more focused on one particular disease, or how they can dictate sort of the way that we do it. It's just we just need to know what. I don't know. That's the part that makes me nervous about having private people or private organizations being responsible for funding.
Right.
So maybe we need to tie our research to products. I mean, I'd be interested to hear how you see the two things gelling together. In my world, it's for curing diseases. In yours, I'd be curious.
Yeah. No, that's a interesting point. For those who are listening, I just want to. Sergey Brin is one of the co-founders with Larry Page of Google, and so that's who Professor Fellows is talking about as far as like with ASAP. But to your question, I take for example Chris Neal, who you know very well, right? And an amazing and a great scientist, and also even more of an amazing human being. And in their lab, they study like naturalistic vision, right?
Mm-hmm.
And so there are gold mines, or I think the results that lab publishes can easily be plugged in to understand video games and how to make more realistic ones, and how to develop virtual reality better, and what are the different natural statistics to sort of perturb. And so I can easily see the Neal Lab having, let's say, a consultant firm, right, almost as an arm of its lab, where they are able to have contracts with like Microsoft and Xbox and all these gaming companies that they would gladly engage. And so that's one aspect of a product. I'll give you another example. There's another amazing scientist at the University of Oregon, Michael Ware, where he studies like audition, right? I can imagine building next generational hearing aids, right? And collaborating on that and understanding what certain frequencies are optimal and things like that. I mean, which we'll get into shortly, you do an amazing and world-class work on retinal waves. And so I wonder, could you think about prostheses, retinal prostheses? And so that I think may be one of the futures of the future scientist, is having to not necessarily be at the forefront of the product, but very much aid in the new generations of products. And so that could be some of the things. For me, for example, I have a company which is called Figbox, where we ask people, hey, like, these naturalistic conversations that you're having, can we take your conversations and put it into scramble the data, scramble your identity, and put it in a repository where only scientists and researchers can learn from those conversations while your identity is protected, and that's open source. And so that allows people that want to study like cultural differences and linguistic patterns, because most people don't know that Professor Fei Li, who is the curator of ImageNet, this database was crucial for understanding like computer vision, right? And so databases are important, and so she contributed to that. And in fact, she has a lab now which is worth $1 billion, right? She's left academia. It's called the World's Labs, where they're building these world models. But so there are different flavors that I think we can start to explore. Also crowdfunding, right? Like that's also another thing, right? There's a site called experiment.com where you can talk about your experience, and the public can fund it and be a part of that process, right? And so these are the different ways I think we can explore. What are your thoughts on that?
Yeah, I mean, I think that sounds fascinating. It's not a world I know, right? And I have learned that the devil is in the details, you know. And this is kind of where I said it would be good, and so someone like me would have to train on, or to learn how you find out about these opportunities and how you match your research to that. I think it's interesting. There certainly is a lot of effort, I know, in disease communities, right, where there are patient groups, right, that often meet with PhD scientists and clinician scientists, but also PhD scientists, so that they so that you have a sense of how to direct your research in a way that might lead to a cure that works for that patient population, right? So I think there's an increase in that kind of conversations that are going on. But for the other examples you gave, it's just not a world I live in. I would be more than happy to go there. That's where my funding comes from. I just need to get educated on how to do that.
Yeah. And, that's an incredibly humbling response. And I think that, some people will use the metaphor that a lot of the labs are effectively startups. When you start a lab, it could be it's akin to like a startup where you're trying to sort of figure out things, and so I think there's a potential there for sure. I want to move into like an even more exciting part of why I wanted to have you on, retinal waves and all of these sorts of like brain activity. So my question related to that is, before we open our eyes, before we see light, for example, right, the brain is already active. What is the brain doing before we are aware of the world? Can you explain to our audience a little bit about that?
Yeah, and so I think by before you mean as an adult human, for vision, when I close my eyes, what's the brain doing? Then how does that change when I open my eyes? Yeah. So it's not a world I know super well, but there definitely are it's a big area in neuroscience to sort of answer that question. And I think I've been very influenced by a scientist that's here at UC Berkeley, whose name is Bruno Olshausen, and he represents a lot of vision scientists, who have this idea that you're carrying around in your brain an internal set of images. Mm-hmm. Right. So when you close your eyes, you're kind of looking inwardly, and you're going through that internal, sort of set of images that are there. And these are images and templates that are built from experience. And I'm going to say your training set, like whatever your training set is, that in the modern parlance. And so it's constantly active in search and sort of cycling through what those images are. And then when you open your eyes, you're doing a kind of template matching. So you're looking to make sense of the world by saying, oh, I know that's a coffee cup, or I know that that's a hat, or, so by doing that kind of template matching. And that's kind of one of the big models that are out there. So I kind of think that your brain is generating these keeping its library kind of circulating right in there.
Right. Interesting. That's quite fascinating. So rather than simply asking, in your work specifically, whether newborn mice can respond to light, your work asks something even deeper, like how does the developing brain distinguish between activities caused by real light and activity generated by internally retinal waves when both produce these bursts of neurofilament? So can you elaborate a little bit about, first, your work, and then kind of like some of the results and questions that are kind of going on in your eyes or in your head, in your mind's eye, in a sense?
Yeah, so I'll start by talking about what retinal waves are. Yeah. And so very early in development, so for humans, this would be, second trimester humans. At this stage of development, the eye and the retina, which is the neural circuit at the back of the eye where your vision begins, it's developed, and it's actually sent its wires, which we call axons, to the brain, and it's made connections to the brain incredibly early. So this is, and that is at a time when there's no visual experience that's going on, right? And it was kind of thought that, and this is true for a lot of your sensory systems, that the organ, like your ears or your eyes, will connect to the brain before vision or hearing is actually happening. And it turns out that during these very early developmental stages, it's not that the retina is silent because there's no light hitting it, but rather it's spontaneously generating this patterned activity, and it's like a little nucleation of activity will happen, and it'll propagate across the retina, and another one will start someplace else and propagate across the retina. And the idea is these are kind of like test patterns that people have shown are propagating up through not just the retina, developing retina, but the connections that the retina is making to the brain. And we found that activity is important for wiring to be organized in such a way that so that when you are born and you do open your eyes, you can make sense of the world, and certainly visual experience will sharpen things up even better, but it's like this sort of gets you in a place where you can be ready to make sense of the world. So that's what we can think of retinal waves. So then there was a discovery about, I guess maybe 20 years ago now that, so my lab and a few labs are really based on this idea that this is the only source of activity that's propagating up through the visual system. But it turns out there was a small population of cells that go from the retina to the brain, but they are also photosensitive, independent. So, the retina is photosensitive because you have these specialized cells called rods and cones.
Right.
And it turns out those rods and cones are the last cells to develop and wire into the retina, and that's why you don't have visual experience. Not only because you're in utero, but because the rods and cones haven't kind of wired up to the cells that take that to the brain.
Right.
But it turns out these cells, the output cells of the retina that take that information to the brain, there's about 2% of them that can actually absorb photons and generate a light response. And it turns out they can actually drive a behavior. So you can take so we do all of our experiments in mice. Mice are born at what would be the equivalent of a late second trimester human. So they're born with their photoreceptors not hooked up to the rest of the retina, and I lost my train of thought.
Sorry,
confetti.
I had to. You all can't see this, but there was confetti that came through the screen, so Professor Fellow was a little distracted. Sorry about that. So the second trimester of mice.
My apologies. Anyway, it turns out, so we didn't, it turns out these cells are there, and they mediate this behavior. When a mouse pup, when it doesn't have functional photoreceptors, it will see a bright light, and it will turn away from that bright light. And we couldn't figure out how can it turn away from that bright light if it doesn't have any photoreceptors. Mm. And it turns out it's because there are these 2% of these cells that themselves are light sensitive.
Wow.
And so that led to this question, well, if there's a light sensitive cell, that when a light is present, it can turn away. But then there's also these waves that are making the cells fire all the time. Why are they not turning away from the waves? Like, how do they separate the test pattern from the light pattern? And the answer we found to that is that they are the only cell in the output cell of the retina that does not participate in waves. They have created an independent circuit. It's almost like an independent channel that's there very early. So the rest of the retina is getting wired up to mediate kind of your form, vision, and your ability to read and to, I guess for mice to, as Chris would say, track crickets and things like that. But there is this small population of cells that has a totally independent channel. It goes to a different part of the brain, and it mediates this fear of light. And so for mice, they need it because if they come out of the nest, which is normally dark, they actually emit an ultrasonic vocalization, and the mom will come out and pick them up and bring them back to the nest. So it serves a purpose.
Fascinating.
Yeah.
So it's like there's a local circuit, and then there's this sort of network-wide propagation of these waves that are there to sort of sculpt the architecture and the structure of the system. But at the same time, you sort of want to make sure that you're alive for that future structure. So you have this sort of local circuit just warning you in case, wow, that is just absolutely mind-blowing. When you are thinking about, melanopsins, do they play a role in any of this sort of, and correct me if I'm wrong, melanopsins are the thing that sort of regulates your circadian rhythm, right? Is that a correct way to think about it?
Absolutely. Yeah, so the reason some cells are photosensitive is they have these proteins that are called opsins, and the ones that are in your photoreceptors are called cone opsin and rod opsin for the rods and cones. And it turns out this small population of cells I'm talking about that's there, they have a different opsin that's called melanopsin, and that's the one you were talking about. And it's that same population of cells. And it was actually discovered that long before we were working on this development question, exactly what you're saying, that these are the cells that project to a part of the brain called the suprachiasmatic nucleus, and that's what photoentrains your circadian rhythm. So that's what gets you to have your rhythm of your daily rhythm sort of linked up to light, to the time of day, and these are the cells that mediate that. Yes, exactly. So it's those cells that we're talking about.
Wow, that is absolutely amazing. So when you think about the next frontier of your field, but also neuroscience, like what are some of the questions that you think should be explored? I mean, what is the next frontier of visual neuroscience, or just neuroscience in general, or computational neuroscience? Like, if you were to give me a bucket list, like what is something that you want to see explored even deeper?
Yeah, I mean, I think so there's there's a lot of mysteries that have to do with development. There's a lot of mysteries that have to do with vision. So that was a very broad question. So let me take a moment to think about this. No, that's fine. That's fine. So I have, I know for sort of what I think my lab can handle that can fit into this. I do think there is this interesting idea in general about how good we are at seeing things and actually pulling out patterns from very noisy backgrounds, and why it's so hard to train AI models to do this, right? Computer vision has been really challenging, although apparently in the last few years it's gotten very good. And I personally don't know what has been the technological breakthrough, but I think this idea, sort of the question we had before about how much does the input from the eyes up to the brain versus this internal model, right, of what we expect to see, where do they meet in the middle? Who's more important under what situation? How do those two signals integrate in order to give us sort of the ability to make sense of the world, right? If you think about how many visual stimuli you're getting all the time, right, it seems crazy. So this idea that you can ignore things that maybe you expect and you just pop out the things that are unexpected is a great idea, but I'd love to know where that happens and how to design experiments to test that. Right. I think it's a big question in vision.
Right. That's something that I definitely am interested in and actively research with some of the work that I am doing now. And it sounds like, at least the little that I hope I'll be able to contribute is sort of the computations, right, that are being performed, right? There's sort of either top down or bottom up dynamics, or kind of these local circuits. One of the reasons why I find the work that you do and fascinating is what I like to refer to, and a lot of others, as the intrinsic dynamics of the system. And, you also mentioned something very briefly about how experience sharpens that intrinsic, output, if you may, right? And so, what hopefully we will publish very soon is that a lot of, well, maybe I shouldn't get there, but I'm very fascinated with that. But what I will say is this, that, there are those such as Chris Neal and others that are now exploring animals in the naturalistic environments, which adds another layer of complexity. Do you think that is going to offer new sets of intuitions and results? And is that already adding more complexity to a system that we already can't understand when it's head fixed or stationary, right? Like, how do you see those two worlds informing each other?
Yeah, I mean, I love the work that comes from this lab, and I actually want to link it to the retinal wave story. But I think we have to go there, because I think everything that we're doing, maybe in a head fixed mouse or in my case, we actually dissect retinas from mice and put them in a dish, right? So we're really, they are definitely not, and we can project a visual stimulus onto them that mimics what is their natural experience in order to figure out how the circuits extract features, except we need to know what the natural stimulus is. And that's going to depend on how the animal runs and how it moves its eyes and what are its reflexes. It's like ocular motor reflexes. It moves its head. How do its eyes compensate for it? And these are all things that Chris is getting at. So one of the things I think that's interesting to link, this all gets set up in development. A lot of it is set up in development. And one of the features of retinal waves that we've been exploring a lot recently is the fact that retinal waves actually have a bias in which direction they propagate. And the way that they propagate on the retina is to mimic optic flow. So when an animal moves through the world, there's just a constant stream of images, right, that are going by its retina. There are some reflexive eye movements to try to stabilize objects within that field. Wow. But there is this flow. And it turns out waves, you have many more waves that propagate so nasally on the retina, lens inverts it, so it's mimicking optic flow. Right. And the idea is for mammals at this stage of development, they're just stationary. In humans, they're really stationary, they're in utero. But for frogs and fish, they're swimming at this stage of development, so they have constant optic flow. Right. And so this idea that retinal waves are a way to generate optic flow, and how important optic flow is for setting up these internal circuits to help you navigate, that is super interesting, and I can't wait to talk to you and Chris about this and show you our data, because I think that there might be some interesting intersections there. And there's a lot of information coming in on the retina. How your brain extracts what's relevant and what isn't is the big question. Yeah.
Wow, that's I'm so excited. I can't wait for you to share it. One of the things that I really admire about you is not just your science, but your humanity. A lot of people don't know, but Professor Fellow actually influenced the current trajectory I'm on right now in the current lab that I'm in when I sort of left Genelia. And, when you were, and so that begs the question, right? Like, your mentors, right? Like, were they people that prioritized being human first and scientist second, right? Were they people that instilled in you the importance of being a great person as well as being a great scientist, right? Where did that come from, or was it your parents that instilled that in you? Could you share a little bit of that? Why is it important to be kind in general? I feel like when I sort of ask or perturb PhD communities around the world or postdoc communities, I think the net feeling is not that feeling of I am important or I feel appreciated or this experience has been great, right? And maybe I'm speaking to the wrong people, right? But and so I do find, or at least I find people like yourself and Chris Neal to be salient objects, right? Not to say that most scientists are not, right? I haven't met all scientists, but I guess where does that humanity for you come in, and how would you sort of, how would you rationalize where the origins of that?
Yeah, I don't know. I mean, I'm gonna probably give a less. I don't know. I mean, I would say that I come not from a family of scientists. I come from a working class background. I had zero expectations that I would be living the life that I'm living right now. I mean, and so it's it's hard to know where that comes from, except for maybe it comes from there. But I'm going to counter it with like a science argument. And I don't know Chris that well, but I would say that, one thing that he and I have talked about is I don't feel like we're being particularly nice. We're just finding good scientists. So we think that there's a lot of really good scientists out there. We want them as our colleagues, and I think that people without any vision of what that path is on how to get there are at a disadvantage, but it doesn't mean they're not good scientists. And so we like to find those, so we're always looking for them. And so I don't think I do, I would never just want to be nice to someone and say, oh, you want to be a scientist, let me help you do that. I mean, I do that. I'm an educator. I do that all the time. But in a case, like you or other people that I mentored, there are great problems and you have a great skill set to work on. We just got to get you in the right environment. So I feel like it's more like a professional matchmaker, although that also sounds bad. So I think that is to try to ignore, the pedigrees or the, something just to meet the person where they are as a scientist. And one of the wonderful things about neuroscience is there's so many hugely unsolved mysteries. Yeah. And it sounds like even you're right where it's like we don't even know how to ask the questions. Right. So how can we answer them if we don't even know how to ask the questions? It's like an all hands on deck. You have a very novel way of looking at things. Right. Chris has a novel way of thinking. Well, and he also has the technical ability to take, crazy ideas on how, what's the right. There's a right way to test this hypothesis. You're talking about how does natural vision affect, the firing patterns in the brain. Right. But you got to be able to do that, right? And so Chris is very good at actually doing that. Definitely. And so, and some of us are a little more limited in our ability to implement that. And so we kind of meet the science where it is. Right. And I think that so I think I'm good at that. And so I like to help people in that way.
So I think that's an interesting way to answer the question, right? Like it's like how do you match the right people together in ways where their skill sets can complement each other to answer or query very tough questions. And and so perhaps I was just going to say there's also a human side, which is I think I know what it feels like to want to do science, right? And it's really hard to do it doing anything else when that's what you want to do. Right. And so I want to try to help. That perhaps is the human side, is to help people pursue that dream for as long as they want to pursue it. So but yes, in addition to solving the hard problems. Right. So I guess one of the things, what your definition of a good science is, that desire, that passion, right? And that curiosity, right? And then also the component of being able to have the skill sets or at least work at it. And then, the two sort of come together in a way where it fits so almost like a glove. And so, it's, another thing that I've gotten from this conversation is how much kind of self-teaching or self-discovery one sort of how much I feel like when I go through the journey as a scientist, there's a lot of things that I don't know, right? And my willingness to learn those things are important, and my willingness to admit or find the right people that can help me learn those things are important. And so I guess I get the sense that when I speak to you, or I get the feeling that I can sort of articulate to you the things that I don't know and ask the right questions to get help. And so hopefully people listening, or the PhD students listening, and the postdoc listening, and scientists listening, is, don't be afraid to ask for help, to reach out. There are great scientists like Professor Feller and Chris Neal that are willing to sort of help and steer you in the right direction. I have two more questions, and then we can wrap it up. The question really circles back to scientific funding, or should we be thinking about a different publication paradigm, and a different way to think about the merit system in science. So for those who don't know, most scientists, we pay to get our work published. And sometimes when we review papers, we most of the time do it for free out of the goodwill. So we effectively pay to publish, and then we pay to access our own publications. And so there's a lot of different legacy components that make it hard. And then, of course, right, this notion of publish or perish, right, is also a thing. What would you say to all of that? I mean, it's a hard problem, right? Like eLife journal funded by the HHMI, the Harvard Youth Medical Institute, tried to solve that, but ended up regressing back to the mean. Do you think there's a solution? I mean, of course, it's a very hard question and unfair in some sense, but what is your general take on all of that?
Yeah, I mean, I think so. Journals do a service. They do find reviewers. They do mediate that process. So I think having, I think peer review is absolutely important. I don't buy what is the current eLife model that, when the author says it's ready to go, then they should just publish it, and then we'll just find reviewers and they can just comment on it. I do think there has to be some curation. There's just too much going on out there. The fields that do that well, like in math and fields, it's just not as big as our field. Yeah. And so I think there's just too much happening to not have some level of curation, or we're all just going to go insane, right? So I do think they do a service, but the hierarchy of journals is a little maddening, and that they are able, I kind of like my lab tries to publish as much as we can in the Journal of Neuroscience, because that's a Society for Neuroscience-based journal. We're helping our community. It's a not-for-profit, right? But somehow one of the big giants like Elsevier has really taken a lot of those papers that used to be in Journal of Neuroscience, and somehow we have allowed them to create a whole series of journals that we somehow think are more impactful than Journal of Neuroscience, and so it's like those resources have gone over to them, right. and it's not that Elsevier is evil, although people have made that argument. It's just that they have a very successful business model, right? And we are letting them continue to be successful. So I don't think it can be a free for all. We are in a huge transition right now because of AI, I think, and there are a couple kind of reviewing or editorial based environments out there that I think are fantastic, and I think that is what's going to change everything, right? So I think that there might be some way to use AI to help do the evaluation of some papers, but I don't know, right? I have seen I'm part of this study. I know I've looked at these AI generated reviews versus what I would do, and they do great. They do great. But I know they do great because I've spent several decades reviewing papers, and so I can. And I don't know what it means if people are trained. If that becomes the norm, and you don't have a lot of people around who have done a lot of reviewing before. So I feel like we're in this transition right now, and I just don't know what to make of it. That's
a very fascinating use case, actually. I never thought about that, where you train a model on an expert reviewer, or you have an expert reviewer grade the model's results over some duration. And then that model will know, okay, in your domain of expertise, let's say in retinal physiology and retinal neuroscience, this is what you look for, right? And somehow integrate that review process, or integrate that into the review process. What I haven't worked out, because this idea just, you just incepted this idea in my mind, is more of, okay, so now as a scientist, I can now do more science. I don't have to do as much reviews. And then will that drive the cost of operations such that the journals, right, aren't interested in making as much money. But I often get the sense that, especially, so I'll give you a very small example of one of the things that my company is working on. We are calling it office hours. And so the idea is, we're building for a VC in Atlanta right now, who was quite successful in a lot of big companies that people use. And the idea is that, he has a lot of people that wants to ask him questions about startups and things like that. And so now you'll be able to sort of speak to him 24/7. And that, so he gets less inbox emails, right? Because he's having to ask or answer the same question all the time. And so if he can not have to repeat himself and then spend more time doing what he likes, that'd be a net positive. So I do agree that there's a transition period, and there's a lot of different ideas, but ultimately it's about how good these models can be and the execution of things. My last question, which is a much more poetic question. How do you? You said not your specialties.
Not my specialty, but I'll try.
How do you want your work to be remembered, and how do you want people to remember you? Like, how do you think about that? And the reason why that question is so dear to my heart is because, when I reached out to you, that was a very critical moment in my life because my father had just passed away. And, it was almost like that slowly became the dominant thought in my mind. And it still is, right? Hence why I'm asking it. And, this idea of, are you doing, it's very much sounds like you're doing what you love, right? Which is great. But it's like you also, at least for me, right? One of the things that I shamefully will admit is I want to be appreciated, right?
Yeah.
And so that's really the origins of that question. Like, how do you want to be remembered as a person? How, like, and are you fulfilled in both your work and just life in general, right? Like, was science a great path? Was it a great choice looking back?
Yeah, it's a great, I can't believe it's lasted this long. Like, it looks like I'm going to make it to retirement as a scientist. And then, pretty much at I think people, maybe it's a form of imposter syndrome, maybe it's a form of reality, but I just kept thinking it's going to end at any point. And given the current climate, it really might end at any point. It's feeling a little risky right now that the party might be, it's not really a party, but this career path might be over. I feel so privileged that I've been able to pursue these scientific ideas that I have found interesting. To me, one of the greatest, I love teaching, so teaching at a big university like this is something that I also really enjoy, and I think I've had an impact. Sometimes I wonder if that impact is greater in the classroom than it is in the research environment. But I think what I have learned, I've been around long enough in the research environment, and I've seen some really great scientists who have passed away and are very quickly forgotten. I will not be remembered. I will not be remembered for the science that I've done. I think it was shocking to me to learn how people who were so big, and then when you're gone, you're gone. People are. It's hard to be remembered, but you get remembered. I guess by interactions like this one.
Definitely. It's those sort of micro behaviors and.
Paying it forward. I just wanted everyone to keep paying it forward.
Yeah, exactly. Yeah, definitely. The things that I remember about when I do a quick scan of the experiences in my life are those moments where no one saw, right? Where that, I remember I was running recently around this area that I live, and as I was running, there was a woman that I was running past, and I waved at her. I didn't know her, but she was a gentle lady, I would say. And as I turned back around, she stopped and just started smiling at me. And that was such a moving moment for me because, I don't know this person, she doesn't know me, but that human exchange was so powerful. I'll never forget it, right?
Oh, I lost your volume.
I said that human exchange was so powerful that, and so, but it was.
Well, I think the other advice I would give you is every victory, every positive review, every paper that gets in, every experiment that works out, celebrate.
Celebrate those small moments, those small wins,
Because that's usually all we get until you get older. Then you get some other things.
Yeah.
But it's you got to celebrate the small ones, because it's mostly what you get is negative stuff. And so you have to celebrate the small ones, feel good about what you're doing, spread some humanity, and it'll get you through the day.
Yeah. I really appreciate, once again, you coming on. It was such a blast speaking with you, and thank you for coming on again. And I can't wait to hear more about your research. Hopefully, you're able to come to the lab and share the retinal waves and the optic flow synergy that you all are exploring. Thank you so much.
All right, thank you so much.
Bye.